The topic of the trial lecture is epigenetics in forensics. I will start with a motivating example, then recap the biological background, including DNA and conventional profiles, the role of epigenetics with some key concepts, and DNA methylation patterns. Then I will move over to how epigenetics can be used in forensics, focusing on age prediction, body fluid identification, and discrimination of monozygotic twins. As we go along, I will explain some examples of differentiation and detection techniques commonly used in forensic epigenetic research. Finally, I will sum up with some concluding remarks. Daytona Beach is located on the east coast of Florida, a bit north of the city Orlando. Apparently, it is the world's most famous beach. It may so be, but bad things have happened here. In December 26, 2005, the body of 45-year-old Laguetta Gunter was found in an alley, shot in the head with a .40 caliber bullet. The police believe she was murdered 2 days before, on Christmas Eve, or the very early hours of Christmas. Foreign DNA was recovered from the body. On January 14, 2006, the 34-year-old and mother of 4, Julie Green, was found in a ditch near a construction site on LPGA Boulevard, also shot in the head with a .40 caliber bullet. Foreign DNA was recovered from the body. A little more than a month after the second killing, in the afternoon of February 24, 2006, 2 people walking in a desolate wooded area off Williamson Boulevard spotted a body. It was 35-year-old Ivana Patton. No DNA was found, but the killer had used the same weapon in all 3 shootings, a Smith & Wesson .40 caliber Sigma series. Days after Patton was found, police announced they were looking for a serial killer. About 2 years later, in January 2, 2008, 30-year-old Stacey Gage was found shot to death in a wooded area next to an old church. There was no foreign DNA on her remains because she had likely been killed about a month earlier and had been exposed to the elements. The police said that this killing was almost a carbon copy of the other 3 women. The suspected serial killer was dubbed the Daytona Beach Killer. What have the police done so far to try to solve the case? There was no match between the unknown DNA profile obtained from the victims and persons in the national DNA database. The police felt taunted by their killer because the man left his DNA behind, knowing that it would not be found in FBI's national DNA database. 2 suspects, including David Lindsay, convicted of killing his wife, was ruled out thanks to DNA analysis that did not match the profile recovered from the victims. The police had been hoping for investigative leads through familial searching that could identify a close relative to the killer. It was still not legal in the state of Florida in May 2011, but have since become legal. I did not find any sources, but we must assume that the search did not help the police narrow down a suspect. Let's take a step back and refresh our knowledge on conventional DNA profiling and the biology behind. The human DNA or genome is divided into 23 parts called chromosomes. The human egg and sperm has a single copy of each chromosome, and when the egg is fertilized, they combine to 23 chromosome pairs, a total of 46 chromosomes. The fertilized egg divides and specializes into all other cell types that make up a human person. These cells all have 2 copies of the DNA. The chromosomes are numbered according to size, starting with the largest. The smallest chromosome pairs X and Y determine the gender of the person, in this case, a male. The egg cell is the largest human cell, and the size is about the same as the diameter of a human hair, but a typical cell is perhaps 10 times smaller. The human genome consists of a sequence of about 3 billion nucleotides. If stretched out, the DNA is approximately 2 meters long, so to fit inside the tiny cell nucleus, the DNA is highly compacted at multiple levels. For example, the DNA strand is wrapped around protein complexes to form nucleosomes. The sequence of the nucleotides G, C, A, and T can code for proteins and are then called genes. The nucleotides form pairs of cytosine and guanine and thymine and adenine. Today's forensic DNA profiling assays look at special places in the DNA called short tandem repeats, or STRs. The so-called STR markers are located outside of the coding regions of genes. The DNA analysis can therefore not tell us anything personal, like for example, predisposition to diseases. The only information retrieved is a numerical DNA profile that can be used to identify the donor of a crime stain by comparison to a known reference profile. The numbers are derived from the number of repeated DNA patterns. In this example, at marker 1, the AGAT pattern is repeated 7 times, and at marker 2, the GATA pattern is repeated 8 times. The suspect has 7 repeats in marker 1 and 13 in marker 2, so someone other than the suspect is the donor of the crime scene DNA. As a consequence of different number of repeats in one marker, the length of the DNA sequence is different. These variants are called alleles. A complete DNA profile of today in Norway consists of 16 different STR markers, plus a marker for gender determination to tell if the person is a male or female. The XY peak indicates a male. To enable analysis of this many markers on capillary electrophoresis instruments, the markers need to be separated by different dyes. Here blue, green, yellow, that is shown as black for visibility, and red. The allele or number of repeats are indicated by the first number in the label. The height of the allele peak is given as the second number and reflects the amount of DNA in combination with the amplification efficiency for the marker. Short DNA fragments are to the left and longer to the right. The markers with the detected alleles make up the DNA profile that is stored in searchable criminal databases. Remember, conventional DNA profiles represent length differences at multiple STR markers in the DNA. Markers are located outside genes and does not reveal sensitive information. Identification requires comparison between the unknown crime scene profile and unknown person profile. A match is usually extremely certain. Back to the Daytona serial killer. The DNA profile recovered from the victims remain unidentified. What more can the police do with the DNA? Well, there is more to DNA than just the DNA sequence. There is an ongoing research efforts around the world to exploit epigenetics in forensic analysis to harvest investigative leads. If you Google epigenetics, this is the definition you get. The study of changes in organisms caused by modifications of gene expression, rather than alteration of the genetic code itself. I will explain with some examples. This is a representation of the chromosomes in the human genome. Chromosome 1 to 22 and XY if you are a male or XX if you are a female. The DNA sequence in all your cells are identical. It is your blueprint. But you have many cells, vastly different. For example, muscle cells and nerve cells. It is easy to see the need for further regulation. And epigenetics literally means above or on top of genetics. It refers to external modifications to DNA that turn genes on or off. These modifications do not change the DNA sequence, but instead they affect how cells read genes. Epigenetics tell a nerve cell what it needs to know to be a nerve cell. For example, focus on this, and this part of the blueprint, as indicated by the arrows, and you don't need to know that or that, as indicated by the crosses. One of many epigenetic modifications is DNA methylation. A good example to illustrate DNA methylation is the agouti mice. These two mice are identical twins, which means they have the exact same DNA sequence at birth. So why do they look so different? DNA methylation at the agouti gene promoter has shut down the gene's function in the brown mouse. The amount of DNA methylation at each allele is partly random and partly dependent upon maternal nutrition and environmental exposure during early development. A central concept in epigenetics is CpG islands. Here we have a representation of the DNA. The DNA bases are represented as the colored A, T, C, and G structures. Due to the hydrogen bonding, A pairs with T with 2 hydrogen bonds, and C pairs with G with 3 hydrogen bonds. The bases are covalently bound to the sugar phosphate backbone, with sugars marked in green and phosphate in yellow. By convention, a single strand of DNA sequences are written in a 5' to 3' direction. The prime number refers to the carbon atoms in red, starting with 1', where the sugar binds to the base, and at the 3' carbon, the sugar binds with the next phosphate group. And at the 5' carbon, it binds with its own phosphate group. So this is the 3' end at the bottom, and the 5' end is to the right. CG base pair are hydrogen bonded to each other, but the bases belong to different strands of the DNA. CpG dinucleotides belong to the same strand of DNA. CpG stands for cytosine phosphate guanine. So you can see the difference here. CG base pair and CpG dinucleotide. Taking a look at this figure again, to the right, CpG islands are roughly defined as stretches of DNA of 300 to 3000 base pairs with more than 55% GC content. Most of mammalian promoters have CpG islands controlling their expression. Generally, unmethylated CpG islands turn the gene on, while methylated CpG islands turn the gene off. A gene can also have a partly methylated CpG island, working more like a dimmer as compared to a switch. Chemically, the methylation occurs at the 5th carbon atom of this base cytosine, forming 5-methylcytosine. DNA methylation patterns may change over the course of a lifetime. Environmental factors such as diet, even your mother's diet when she was pregnant with you, may influence the level of DNA methylation within cells. Pathogen exposure, lifestyle, and aging are other factors influencing DNA methylation. Forensic scientists have quite recently become interested in epigenetic DNA methylation patterns. In a recent paper, the authors envisioned many questions that forensic epigenetics may be able to answer in the future in order to get investigative leads for the police. Epigenetic forensic analysis may be able to predict lifestyle choices like smoking, alcohol intake, and physical activity, and externally visible traits such as body shape, to mention a few. The 3 most researched and important questions up until today are body fluid determination, age estimation, and discrimination of identical twins. So how could epigenetics help solve the case of the Daytona Beach killer? The DNA profiles were obtained from the analysis of semen, so the police already know the body fluid in this case. However, epigenetic age prediction from semen has been demonstrated in a study from 2015. The result from analyzing 3 selected epigenetic markers had a mean absolute deviation of approximately 5 years from the actual chronological age. So knowing if the killer is young, middle-aged, or old may help the police narrow down the pool of possible suspects. In the future, other characteristics like smoking, alcohol habits, and body shape may be predicted to further homing in on the killer. Remember that epigenetics modifies the DNA but not the DNA sequence. Epigenetics can turn on or off or dim genes. Epigenetics is influenced by external factors like environment and food. DNA methylation is an epigenetic modification. The cytosine in CpG dinucleotides is unmethylated or methylated. CpG dinucleotides are on the same DNA strand, while CG base pairs are on different DNA strands. Forensic genetics are interested in DNA methylation patterns for the possibility of investigative leads and indication on the activity. The Daytona Beach killer was an example of how age prediction may help the police. Body fluid identification is another important challenge. Let's say we find a kitchen knife at the scene of a murder. Trace DNA is secured from the handle and from the blade, and two different DNA profiles are generated. There is no visible blood on the knife. The DNA profiles match the suspect, Sideshow Bob, and the victim, Bart. The conventional DNA profile can with extreme certainty identify the donor of biological traces found at a crime scene. But it cannot say anything about when this trace was deposited or the type of body fluid the DNA came from. Therefore, the relevance to the criminal activity is often the key question in court. The suspect, Sideshow Bob, claimed having dinner with Bart some days prior to the murder, and so had access to the crime scene. Sideshow Bob also claimed helping Bart chop some vegetables for the dinner, so the DNA could have been deposited earlier by activities unrelated to the crime. Identifying the body fluid found on the evidence can give important insights into the activity leading to its deposition. Blood may indicate injury, and such information may strengthen the case against the suspect. Other examples are saliva that may indicate licking and semen indicating sexual activity. Many body fluid identification methods exist today, such as chemical, immunological, and enzymatic tests. However, these conventional body fluid identification methods are mostly presumptive and are carried out for only one body fluid at a time. Therefore, the use of a DNA methylation-based assay would be better, providing a direct link between the recovered DNA profile and its source. Assays for methylation analysis and detection is a jungle of methods. This, now probably incomplete, overview from 2012 lists around 40 different methods grouped into three main strategies based on the underlying biochemical principle. Bisulfite-based strategies. The bisulfite reaction was actually described already in 1970. And restriction enzyme-based. A restriction enzyme-based method was first used in 1979. And the third group, affinity-based, first used in 1985. And there are also some combinations in between these groups. So although some principles are quite old, there have been a continuous development of biochemistry and technology, making it practically suitable just recently. Any methylation assay is basically carried out in two steps. First, one has to differentiate between methylated and unmethylated DNA using one of the three biochemical principles. Here they are depicted very simplified. A. Affinity-based strategies, using, for example, antibodies directed to the methylated cytosine. Restriction enzyme-based, using methyl-sensitive restriction enzyme to specifically cut methylated or unmethylated sites. And C. Bisulfite-based, where the unmethylated CpGs are changed into uracil, while methylated CpGs are protected. Second, one has to detect these DNA sites. Both methyl-sensitive restriction enzyme-based methods and bisulfite-based methods are currently used in forensic epigenetic research. Methylation-sensitive restriction enzyme-based fragment analysis has been one of the most frequently applied methods in forensic epigenetic body fluid identification papers. Provided that a suitable enzyme is available for the CpG sites of interest, this method is very simple. The most used enzyme today is the HAH1, which recognizes the unmethylated target sequence, GCGC. Adding the enzyme to the DNA sample will cut the unmethylated target sites. If the CpGs are methylated, the enzyme does not recognize the site, and the DNA is not cut. After treatment with the enzyme, the sample can be PCR amplified. When primers bind to the unmethylated target, there will be no PCR product, since the DNA has been cut in between the primer binding sites. The methylated target gets amplified since the DNA is still intact. The end result can be detected using genetic analyzers, one of the most common instruments in forensic laboratories. And this is a big advantage of the method. Unmethylated sites will not show a peak, while methylated sites will generate a peak. In a paper published in 2016, this method was used to build a multiplex panel of 8 body fluid specific markers and 2 controls. DC is a digestive control for the enzyme, and PC is a PCR positive control. 2 markers for venous blood, semen, and saliva, and 1 for vaginal fluid and menstrual blood. Here the multiplex was tested on blood, indicated by A, and semen, indicated by B, with robust results down to 250 picograms of input DNA. A drawback with the MSRE PCR-based methods is that they can only be used for the on-off type of CpG sites, which may be difficult to identify. The body fluid multiplex was validated and demonstrated to work well integrated with the commercial STR-DNA profiling kit Minifiler. After adding the multiplex enzyme mix together with the Minifiler mix, the only additional step was a 30 minutes digestion step before the first cycle of PCR. This integration ties the DNA profile to a specific body fluid, which is of great value to the investigation. Remember that body fluid identification is important to strengthen the case. There are 3 main strategies to differentiate between methylated and unmethylated CpG sites: affinity-based, restriction enzyme-based, and bisulfite-based. Methyl-sensitive restriction enzyme PCR-based methods can be integrated into the current forensic genetics workflow with minimal changes. The Terminator 2 Judgment Day from 1991 is still one of my favorite movies. It became famous for its incredible computer graphics of a liquid metal robot. Here the robot mimicked 2 of its victims. It is not widely known, but despite being the most expensive film ever made up to that point, the director James Cameron saved money using identical twins for these shots, instead of using expensive computer graphics. If this was real life, with no liquid robot, the obvious question would be, who killed who? Epigenetics is able to answer that question. Let's first go through the biology. Identical or monozygotic twins are created by a single fertilized egg that somehow is split into 2 that continue with the normal development. The exact cause for the splitting of a zygote or embryo is not known. The birth rate of monozygotic twins are uniform, about 3 in every 1,000 worldwide, or about 0.3% of the world population. Fraternal or dizygotic twins come from 2 individually fertilized eggs, and are no more similar than other siblings. In contrast to monozygotic twins, the birth rate of dizygotic twins ranges from about 6 per 1,000 births in Japan to almost 50 per 1,000 for the Yoruba people in Nigeria. Because monozygotic twins share almost identical DNA, they can't be distinguished by conventional STR DNA profiling. Given a world population of about 7.7 billion people, there are about 23 million identical twins out there, so the problem is relevant, and this is where epigenetics can help. Remember that epigenetic methylation patterns change over time due to environment and other factors. Twins will therefore accumulate methylation differences as they become older. And here is an analysis of twins when 3 years old and 50 years old. the number of detected differences has increased significantly. A recent paper published in May is perhaps the first study addressing epigenetic discrimination of identical twins in the forensic setting, using buccal swabs, saliva samples, and used cigarette butts. After genome-wide studies to identify candidate differentially methylated sites, they applied a targeted DNA methylation analysis using a TaqMan-based, highly sensitive qPCR technique. The initial step is to extract the DNA from the buccal swabs and cigarette butts, of course in separate tubes. Then follows a bisulfite conversion. Bisulfite conversion, or treatment of DNA, is required for many methods to analyze methylation. What the bisulfite treatment does is to change any unmethylated Cs into uracil bases. This includes any Cs within and outside of CpG sites. The methylated C bases are resistant to this change and remain as a C after bisulfite treatment. TaqMan probes were designed to bind to the methylated CpG targets and will not bind if there is a U instead of a C. PCR primers, indicated by the arrows, were designed to bind to either side of the target site. A TaqMan probe is a short complementary DNA sequence that match the target DNA sequence. The probe has a fluorophore attached to one end and a quencher to the other end. When the fluorophore and quencher are close to each other, any light energy emitted from the fluorophore is absorbed by the quencher. Consequently, there is no visible light signal. The DNA polymerase binds to the primer and starts copying the DNA strand. When the DNA polymerase encounters a bound TaqMan probe, the nuclease activity of the polymerase destroys the probe and releasing the fluorophore from the quencher. The fluorophore can now emit light that is detected by the instrument. Real-time PCR detects light after each PCR cycle. During the first cycles, the light is too faint to be detected. As the PCR progresses and the number of targets amplify, the light becomes detectable. The intensity is on the y-axis in log scale, and the cycle number is on the x-axis. A repetitive alu element was used as an internal control for normalizing the amount of DNA input of the target. The unknown DNA samples were compared to standards with varying DNA concentrations and methylation degrees. The sensitivity of the assay is comparable with conventional STR-DNA profiling assays, around 1 nanogram of input DNA. Remember that differentiation of monozygotic twins is an important question in some cases, because their conventional DNA profiles are identical. Bisulfite treatment converts the methylation into a change in DNA sequence, which enable detection using DNA sequence-based methods. No robust and easy-to-implement method to differentiate monozygotic twins exists today. Finally, before the concluding remarks, I want to show a short animation of yet another technique for methylation detection. Back in around the year 2000, when I was studying biotechnology at the university, single molecule sequencing was a theoretical possibility, but still science fiction. Today, it is commercialized, which is really cool. The animation shows reading of the genetic code, but the technology can also detect the DNA methylation state. Currently, the error rate is higher than traditional methods, but the technology continues to improve.
At the heart of the technology is a protein nanopore. This model shows a typical nanopore. The core of the protein is a hollow tube, a few nanometers in diameter. Oxford Nanopore Technologies designs and manufactures bespoke nanopore structures for a range of applications. In nature, nanopores form holes in membranes. In Oxford Nanopore's technology, the specifically designed nanopore is inserted into a membrane created by a synthetic polymer. This membrane has very high electrical resistance. By applying a potential across the membrane, which is bathed in electrophysiological solution, an ionic current can be generated through the nanopore. Single molecules that enter the nanopore cause characteristic disruptions in the current. This is the nanopore signal. By measuring the disruption, DNA, RNA, or protein molecules can be characterized.
The DNA strands to be sequenced are mixed with copies of a processive enzyme. As the DNA enzyme complexes approach the nanopore, the single-stranded DNA is pulled through the aperture of the nanopore. The enzyme, shown here in green, is designed to ratchet the DNA strand through the nanopore one base at a time. The enzyme binds to a single-stranded leader at the end of the double-stranded DNA template and unzips the double strand, feeding it through the nanopore. As the DNA moves through the pore, the combination of nucleotides, called a chimer, within the narrowest part of the pore barrel creates a characteristic disruption in the electrical current. This information can be used to determine the order of the bases on that DNA strand.
Using microchip fabrication techniques, Oxford Nanopore has developed highly scalable arrays of these nanopores to go into devices of any size, from the mobile phone-compatible smidge ion to the portable MinION and high-throughput PromethION. These are designed to provide on-demand biological information for any person in any environment.
So, some concluding remarks about epigenetics in forensics. Conventional DNA profiles identifies known donors of a trace with high discriminatory power. Epigenetics can provide the police with investigative leads if the donor is unknown. Epigenetics can identify the body fluid of a trace, implying the activity, which is a key question in court. The possibilities are many, but most applications are currently not mature for forensic use. A broad implementation required the use of existing instrumentation because of economic reasons. Many combinations of biochemical principles and detection technologies exist. Bisulfite treatment changed the unmethylated DNA code, enabling sequence-based detection. Methyl-sensitive restriction enzymes cuts DNA, enabling fragment-based detection. Thank you.